Hello everyone, today I like to guide you through a small exercise in Nomad. We will be modeling a simple cell. Since this is just a practice project, we won't focus too much on structural proportions. So first, we'll create a basic sphere, scale it up to serve as a cell membrane. And later, we'll add some textures on the membrane to give it a rough appearance. Let's first set a camera so that we can easily switch to this view later when we do the rendering. Now, let's adjust the shape of the cell membrane, making it more elliptical. For sculpting the texture, I'm using a brush to create some uneven volumes on the surface. You'll notice that symmetry is enabled in the top left corner, which saves some effort. I've chosen XYZ axis symmetry, so painting only 1/8 of the area is enough. The difference between local and world symmetry is that they behave the same when the object's center coincides with the world center. However, if the object is offset from the world center, there will be some differences. Okay, so now the cell membrane is complete. To avoid it obscuring the internal structure, I reduced its opacity. And next, we'll build the nucleus and nucleolus. Since I don't plan to add nucleus pores, these two parts will be relatively simple. So now we have the basic cell structure with the cell membrane and nucleus. And next we'll add some organelles. So first let's start by building the centrosome. We know that the centrosome consists of two centrioles. Each centriole, for simplicity in this exercise, is made up of 9 units, each consisting of 3 cylinders. By building the centrosome, we can practice using arrays and mirroring, which were covered in the basic lessons. If you've forgotten the theory and the method, you can refer to the previous lesson video. So after assembling these 9 units into one piece, we can apply various transformations to the whole structure, like naming it. Here, I encountered an issue. So you can see that when rotating the structure, it collapsed. Upon inspection, I realized that I had enabled symmetry in the top left corner, which can cause the model to rotate from two directions simultaneously, leading to this issue. The solution is simply to disable the symmetry. And now I can make the two centrioles perpendicular to each other. The centrioles seemed a bit narrow, so I used the axis transform tool to elongate them. And here I'm using the orthographic view. The difference between an orthographic and perspective views is that while perspective can give us a better simulation of real-world effects, it can sometimes distort our judgment of length and spatial relationships. The first time we encounter an orthographic view might be in the elementary school, with grids resembling those, how do you pronounce it? Parallelograms. I will create a group so that both centrioles can be operated together later. You can also connect the two centrioles directly in this case. And next, let's try to build a Golgi apparatus, which is very important. The primary technique used for this is Nomad's classic feature, which is shell extraction. The process involves selecting the object, or selecting all, and creating the mask, and then going to the operation page to choose the shell extraction tool. And here I just start by creating a sphere, then flatten it, which will be splitted into 5 vesicles. I'll shape each vesicle to make it look more natural. Well, in this operation, I kind of made a mistake. If some of the vesicles are too flat in certain areas, it can affect the subsequent shell extraction, because the principle of shell extraction is like selecting a region in 3D space, starting from the inside and working outward. If certain areas are too thin, it may select parts of the outside. So you'll see that when a surface group is enabled, sometimes the internal faces can flip to the outside. But of course, this doesn't have a major impact on models with low precision, but it can be an issue if you plan to paint different colors on the inside and outside of the object. So it's very important to make adjustments in the early stages of modeling. And now I'm going to restructure the mesh here to make the edges look more smooth, like this. You just redefine the resolution. Well, there are also other ways. Like, you could create 5 separate spheres and flatten them one by one. I'm doing this instead of that is because this may make them appear as one cohesive structure. Because biologically, the vesicles are produced from the Golgi apparatus. So, if we create 5 separate spheres and flatten them, the smoothness, of course, will be better. Here I'm adding some clay around the edges to make the polygon distribution more even and remove those sharp edges. The next step is the shell extraction. So first, you create a mask. Select all the objects you want, and then go to the menu. In the top right corner, I measure the thickness to set the thickness parameter more accurately. The thickness should be negative and shouldn't exceed the thinnest part of the model. Otherwise, it will stick out. And afterward, we use a trimming function to check if the inside is hollowed out. Like this. You can see that it is quite good. So let's repeat the whole process. Let's just speed it up a bit here. After shell extraction, the original model needs to be discarded. So here we have split, auto split, which is a result of the cutting process, and EXT, which is from the shell extraction. And we only need to keep the object obtained from the EXT, and we hide or delete the rest of them. Now let's talk about the use of the tube tool. We can create a tube and add vertices based on the desired curvature. I'm trying to create a microfilament here with the effect of being thinner at both ends and slightly thicker in the middle. And then rename it. Okay, so now that we have most of the elements, we can proceed with the final assembly. At this stage, we need to adjust the proportional relationships between each element. And here I locked the camera to avoid accidentally selecting it. And I adjusted the position of the microfilament. While doing this, I accidentally made one of the microfilaments too small, and it is quite hard to manipulate, so I just delete that one. Okay, now let's create the cross-section effect on the right side. There are actually two ways to do this. The first is to merge all the elements together, which will give a one-step solution, but has the disadvantage that you'll need to manually paint the colors later. Alternatively, we can use surface groups, but they might be a bit tricky for beginners to understand. By the way, I'm also a beginner. So I'm using the method where I just cut things separately. You can see that the goji body wasn't cut perfectly because some shapes weren't done very well earlier. And during the shell extraction, some internal faces may have flipped outward, but it's not a big problem because We only need to export a rendered image. You can always choose a specific angle to cover up any imperfections. Of course, it's not always recommended. And here, since the cytoplasm looks quite empty, I decided to create a few mitochondria. For simplification, I made them into a flatter, elongated ellipsoid with a rough exterior. The process is just the same as with the cell membrane. Then I created a vesicle with some particles inside. Here we can talk about the difference between instancing and cloning. For the mitochondria earlier, I used cloning, and now I'm using instancing. Instancing can be seen as creating multiple identical objects from one mold. When you color one, all the other objects created from the same mold will change color as well. The advantage is that it saves many time. So here I find that I can't use the axis transform tool. Well, here's the reason. I hadn't removed the mask. As you can see, the vesicle's color is darker than the others, which means it's selected by the mask. So I need to click on the mask and deselect it. By clearing it all, the vesicle goes back to its normal white color. After clearing the mask, I proceeded with instancing, and now you can see an identical instance appear. And next, I create a new camera angle because the previous one wasn't very good. Just click Add View to set a new angle. Now let's talk about rendering. As rendering is not the most important part at the early stages, I'll briefly explain it here. All the materials in the scene are applied through the material menu. The most unique one is the cell membrane. In addition to using the coloring tool here, I also applied a material from which you can find from the top left corner. In the entire scene, there's only one light source, which is a point light. The blue glow around the edges of the cell membrane is achieved with ambient light. You can find it in the PBR settings. And in the PBR settings, there's a scene where we can adjust the exposure and rotation to change the appearance of the ambient light. Once everything is set, you can export the image with either a transparent or opaque background, depending on your choice. And that's all I have to cover today. Since rendering wasn't recorded, you can try it yourself or refer to the last video. If you have any questions, feel free to leave it in the comments. As I mentioned earlier, I'm also a beginner, so let's just learn together.